All right, guys, welcome back to the spring semester. I hope you guys had a great break. I know we have started off this unit by introducing the video on DNA technology, kind of the birth of DNA technology, and hopefully you learned a little bit from that. And we'll kind of refer back to that as we go throughout the unit. So, for the most part, this new semester is going to be focused on the bigger scale, but this is the one and only unit that where we're kind of really focusing still kind of on the micro side of biology, and that we're going to be looking at viruses and we're going to be talking about bacteria, and then we're just going to talk about how we can use those in DNA technology. And so, just to be clear, this is why we're covering this here, is because this is where it is really relevant. Now, viruses are going to be our focus for today. And just to kind of fill you in a little bit, they are kind of biological weirdos. They don't really fit the criteria for being a living thing, but yet they obviously are very much a part of our daily lives. They infect all different types of organisms and can disrupt, cells and how they work. And so, in order for us to understand DNA technology, which is kind of the heart of the unit, we need to understand a little bit more about these cells and viruses that we often use in DNA technology. And so, as I mentioned, viruses wreak havoc in pretty much all areas of life. There are viruses that infect bacteria. Obviously, as humans, we have viruses that affect us and cause us to be sick. Animal viruses, plant viruses, there's going to be fungal viruses. They pretty much affect all forms of life. Now, they themselves are not really considered alive, and how they came about is not fully understood or known. Just like with cells and living things, they vary. How they work can vary, their structure can vary, and so those are some of the things that we're going to talk about today. Now, the first thing to note is that they are incredibly tiny. They are smaller even than bacteria. And so we know that bacteria are microscopic, and bacteria are typically measured in micrometers. Micro means 1 millionth. And so bacteria are usually, millionths of meters in size. Well, viruses, as you can see, are measured in nanometers, which is billionths of meters. So they're quite a bit smaller. We have trouble seeing bacteria with light microscopes. We're not going to be able to see viruses with light microscopes at all. The only way we can see them would be with some of those electron microscopes. Now, their basic structure, essentially, a virus is two components, okay? All viruses consist of two main components, and that's going to be some type of nucleic acid. We're going to see that for viruses, one of the things that's different about them and living cells is they're going to have DNA or RNA. And then that nucleic acid is going to be surrounded by protein, and we call that protein coat a capsid. So notice they do not have cellular structure, and that's why we call them acellular. Anytime you put an A or an N in front of something, it means not. Please do not get this word confused with unicellular, which means one cell. Acellular means no cell. It is not a cellular structure. Notice there's no cell membrane, there's no cell wall, they don't have cytoplasm. When we talked about cells, there were four main components. Now, the thing that they do have in common with cells is they do contain nucleic acid. They're going to have DNA or RNA, like I mentioned. They do have proteins. They might, notice that's a might, not all viruses have this. They may have an additional component to their structure, which is called an envelope, and that's going to be made of phospholipids. Now, notice we are not calling it a cell membrane. What these viruses can do is they actually steal some of the phospholipids from the host, and they create this additional layer called the envelope. But it is not a cell membrane, it is not a phospholipid bilayer like what we've got. It does not have all the components that our cell membrane has. Now, they might contain some enzymes, they may have some additional proteins, but for the most part, your viruses are two components: some type of genetic material surrounded by protein, the coat we call a capsid. Now, they can vary widely in their shapes, and that has to do with the proteins that are going to be on the outside. So there's four main groups. We have the filamentous, which this is going to be like your tobacco mosaic virus. This was the first virus that was ever discovered. As its name implies, it affects tobacco plants. And it kind of has this filamentous, when you look at them under the microscope, they almost look like little tiny cigarettes, which I think is a little bit ironic. But they're kind of long, like a straight filament. They could be isometric. Some of our human viruses are this way. They're very geometric-like. They have kind of this multi-sided shape. You have your enveloped viruses, which do have that additional envelope on the outside. A lot of our viruses are enveloped. HIV is enveloped. Your flu virus, your basic cold virus, that's enveloped. And then we have what are called the head and tail, and this is going to be our bacteriophages, and this is a word that we need to get familiar with. Okay, we're going to see bacteriophages quite a bit as we go throughout DNA technology. But a bacteriophage is a virus that infects bacteria, okay? And that head and tail structure is like what you see over here. I always say that they remind me of cookie robots, if you've ever seen Despicable Me. But they kind of have this head-like structure that all of this is made of protein. So I know it looks funny, but all of this is protein on the outside. And then they have this tail that they use to attach to the host. So this is what we're referring to. Here's a bacteriophage here, and here's a bacteriophage here. But once again, on the outside, what we're seeing for the most part are these proteins. All of the little spiky objects are proteins. In the enveloped viruses, there is that phospholipid envelope, but they still have these proteins that are sticking out. And so they do come in different shapes and sizes. Now, we brought up the word proteins, and we talked about proteins a lot first semester. And I just want to remind you that anytime we're talking about proteins, we are always talking about specific. And that same thing is going to apply to the proteins in our viruses. All right, so next we're going to talk about the genomes. And this is another word we need to get comfortable with, because a lot of times, for whatever reason, students kind of miss the main point. Anytime we're talking about genome, we are talking about the genetic material specifically. The second portion of these notes are all about bacterial genomes. So we're going to focus on the DNA and the RNA that you'll find in bacteria. Here we're focusing on the genome, or the DNA, that you'll find in a virus. Now, they do tend to be small. They really don't need a lot of genes, because what a virus is going to do is it is going to use a host cell to make copies. And so they don't need genes to code for all of their proteins and functions and enzymes. They pretty much need a very small number of genes that allows it to utilize the host. So they don't have a ton of genes inside of their DNA or their RNA. Now, once again, an important word here, because if we're talking about a living thing, if we're talking about a bacteria all the way up to a complex animal, living things are going to have both DNA and RNA. But when it comes to viruses, it's going to be one or the other. There's DNA viruses and there's RNA viruses. And even when we're talking about their DNA and RNA, it is going to be different from a living thing. If a living thing has DNA, it always is the double helical structure, the sugar phosphate backbone, the nitrogen bases on the inside. But if we are talking about viruses, there could be a single strand of DNA, or there could be a double strand, or it could be linear like we see in eukaryotes. It could be circular like we see in prokaryotes. So viruses are going to be different in that they're going to have DNA or RNA, but that structure could also vary, okay? In a living thing, DNA pretty much always has the same basic structure. Now, the DNA viruses are essentially going to enter into the cell. That genetic material is going to get copied. The genes will be transcribed and translated to make those proteins. Now, if it's an RNA virus, there's going to have to be an extra step, okay? If it's an RNA virus, it's usually going to contain the enzyme reverse transcriptase. And we're going to see this a couple of times this semester. We'll see it in DNA technology. And then we see it here with retroviruses. So reverse transcriptase is pretty much responsible for doing exactly what it says. It is the reverse of transcription. So just to go back to first semester, I know you guys have slept a lot. I know the last thing you thought about over Christmas break was genetics or DNA or anything of that nature. And so transcription is DNA to RNA. And so reverse of that would be RNA to DNA. So if a virus has RNA, and we call these retroviruses, HIV is a retrovirus. It can be a very tough one to track down and treat. It's because there's a lot more mutations that happen with these RNA viruses, because before they can really do a lot in the cell, they need reverse transcriptase to take that RNA and turn it back into DNA. And so there's just a greater room for error and room for mistakes when making copies. So if it's DNA, it can just be copied directly and proteins can be made, but if it's RNA, it does have to be turned back into DNA first. Now, as I mentioned earlier, anytime we're talking about proteins, the word specific is going to be involved. And so viruses are very specific to their host. So the host is going to be the cell that they infect and they use to make new viruses. Now, here's the thing. This is something we need to understand. Viruses are no fun. They hurt us, they cause damage, and obviously if we could get rid of them easily, we would, okay? Now, the thing with viruses is in order for them to be able to infect a cell, there has to be some kind of matchup between the proteins on the virus and the proteins on our cells. Viruses cannot just enter into any cell. The only way they can infect and attack a cell is if that cell has certain proteins on its cell membrane, okay? So it has to be a protein to protein matchup. Now, this is what I need you to understand is that these proteins that we have, they are not intended for the virus. We have all kinds of proteins on our cells. They receive signals like hormones and neurotransmitters and other proteins. We talked about cell communication, and so we know a little bit about proteins and how they work. And so our cells have all these different protein receptors on them, but they're not intended for the virus. Viruses have just found a way to utilize these proteins on our cells. But they cannot infect a cell that they cannot attach to, okay? So a lot of viruses use those glycoproteins. Glyco just means they have a carb structure as well. So they use these proteins to attach to proteins on the host, and it is specific. So this is why we have different types of viruses. Like there may be a respiratory virus. If you have a respiratory virus, you're probably not going to have a horrendous case of diarrhea, okay? If you have some kind of intestinal virus, you may not have a stuffy nose and sneezing. And so the reason we have kind of different effects is because those viruses are only affecting certain parts of our body. Why? It has to do with the protein to protein matchup. So viruses are always going to be specific to their host. It's not like there's one virus that can go around and affect every organism and every cell. Bacteriophages are going to be specific to bacteria. There's animal viruses, there's plant viruses. Now sometimes they can be cross species. It really just depends on what the virus is and the proteins that it's using. Now in order for a virus to do anything, it's going to have to, like I said, it's going to have to attach, and somehow the genetic material of that virus is going to have to be taken inside to the host cell so that it can be copied and proteins can be made, okay? Now as I mentioned, that infection is going to be specific, okay? They can only infect certain species. So some are incredibly, incredibly specific. Some of them only infect certain cells. So like HIV, for instance, the HIV virus specifically attacks helper T cells, which are part of our immune system. They don't affect any other cells in the body. They specifically affect helper T cells. Other viruses have a much wider host range, like rabies, for instance, can affect many different species of mammals. Some are going to be, human specific. Some are going to be cell specific. It's all based on the proteins that we have on our cells, okay? And then how we handle it, our immune response, which genes get copied. So all of these things are going to affect whether or not that virus can use that cell and potentially make new viruses. Now, anytime a virus does enter a cell, anytime a virus does use a host, it is going to damage it. Okay, we know that viruses do cause us problems. And so what can happen is when they use the cell, they could potentially kill the cell. They could cause the cell to lyse. Remember, that means to burst or pop open. They could trigger the cell to undergo apoptosis, which is like that cell suicide. Now, the viruses do not always immediately kill the cell. Sometimes the cell will continue living, but normally it is damaged, and so a lot of times it will not continue to function, and ultimately it will be affected by that virus in a negative way. Now, in terms of symptoms, we get symptoms because of two reasons. Number one, we get symptoms because our cells are being damaged, okay? Our cells are being hurt. So like, for instance, HIV, if they're attacking those helper T cells, that is going to reduce our immunity, which means we're going to be more susceptible to just common infections. Now, other symptoms come just from our simple immune response. We have amazing immune systems that do an excellent job of trying to fight off these pathogens. And so a lot of our symptoms, like the extra mucus and snot, things like that, the fever, all of those are just part of our immune response and our body's trying to fight them off. So that's why viruses make us feel so crummy. Now, how do they actually go through and infect? Okay, there's kind of four main things that have to happen. The virus has to attach. We already mentioned and talked about how there's going to be specific proteins. The proteins on the virus have to bind to receptor proteins on the host. That's always going to be a specific matchup. Once the virus attaches, it's going to have to enter the cell. It's all about getting that DNA or RNA into the cell. Now, sometimes it may just like inject that genetic material directly, so that means it's going to enter without its capsid. It could possibly fuse. The envelope of the virus may fuse with the cell membrane and kind of allows the genetic material to enter, or the cell could actually take it in by endocytosis. Remember, that's kind of the cell kind of engulfs that virus. That would only be triggered, though, by the attachment. So there'd have to be some kind of like a receptor-mediated endocytosis that would have to happen. Now, once the virus gets inside the cell, it's going to have to replicate the genetic material and then make the new proteins for the virus, and new viruses have to be assembled. And so that does depend on whether it's a DNA virus or an RNA virus. Remember that RNA virus is going to require reverse transcriptase. And then once the new viruses are made, they're going to be released. A lot of times this is what can lead to the death of the cell, and then those viruses can go forward and infect other cells around them. And so once again, it has to attach. Genetic material has to get inside of the cell. Essentially, the host cell is going to be taken over. Okay, the host cell does not know that it's making viral genetic material and viral proteins. It's kind of tricked. And so just like it copies its own DNA and makes proteins, it does this for the virus. Once the viruses are made, they have to escape. And a lot of times, or all of the time, they're going to damage the cells in the process, sometimes killing them. Now, two specific viral infections. Okay, I do want to be clear. We're specifically talking about bacteriophages here. We do have similar types of cycles in like human viruses and other species. But there are two main types of bacteriophage infections, and we have the lytic and the lysogenic. Now, what I need you to remember for lytic is that it is immediate takeover. Okay, you might want to write that down on your notes. The lytic cycle is immediate takeover. And so this is where there is very little time between the virus attaching to the cell and the cell dying. Okay, the cell is going to die because it lyses. And so essentially, if we were to relate this to human viruses, these are the type where within a very short period of exposure, you're going to start to see symptoms, okay? Within a day or two after being exposed to someone else that had it, you start to see those symptoms. So pretty much as soon as that virus gets into your body, it's going to take over your cells and it's going to start wreaking havoc, okay? So the lytic cycle is immediate takeover. The bacteriophages attach, they inject their genetic material, they immediately start making new viruses, and then they kill the host by lysing it, okay? So remember that lytic comes from that word lyse, which means to burst open. Now, the second type of cycle is a little bit trickier, okay? Now, I will tell you the end result is ultimately going to be the same. At some point, that bacteria is going to be killed by lysine when the viruses are released. But this one is not immediate, okay? So remember that the longer word takes a longer period of time, okay? The shorter word, immediate, shorter cycle, lysogenic is going to be longer. And so these actually are a little bit trickier in that instead of immediately taking over the cell, the virus's genetic material becomes part of the bacteria's chromosome. So that viral DNA becomes part of the host cell's chromosome. And at this point, we now call that a prophage, okay? So essentially, the viral DNA is just kind of hiding inside of the bacteria. It's not hurting it. It's not making new viruses yet. It's just kind of hanging out. And so it has this latent period where the virus is in the cell, but it's not actually damaging or making new viruses or hurting the cell at this point. Now, to relate this to human viruses, we do have viruses that you could have for a very long time and not necessarily experience symptoms. A lot of your STDs, like herpes, HIV is like this. People could have the HIV virus in them sometimes for months before they have any symptoms or anything that would lead them to believe that they had the virus. And that can be very scary. The virus is just hanging out. Now here's what it does, is because it's in the genetic material of the host, every time the host copies its genetic material and copies the cell, new viruses are going to be copied as well. And so remember here we're talking about bacteriophages, so we're talking about bacteria, but the same thing could happen within us. If we have a virus that is just kind of hiding within our cells, we could be making more and more and more of those cells that contain that virus. Now, at some point, and depending on the environment, at some point, that genetic material will be copied, new viruses will be made, and that cell is going to die by lysing, okay? But that could be several generations later. So normally there's going to be some kind of environmental factor that triggers kind of the change from the lysogenic to the lytic cycle. So essentially you can see the cycles, they go together. The lytic cycle was immediate, so the genetic material enters, new viruses are made, the cell lyses. Now over here, the genetic material enters, it becomes part of that bacteria. Those bacteria can be copied for generations. So 1 becomes 2, 2 becomes 4, 4 becomes 8, and so forth. And then eventually, due to environmental factors, then this bacteria, all of these bacteria that have that virus will go through the lytic cycle. So it can take a lot longer with the lysogenic. All right, we're getting close to being done here. We have a couple more things to talk about. So oncogenic viruses. Now, you might be familiar with this prefix onco. You may have heard of oncology or an oncologist. Onco refers to cancer. And so oncogenic viruses are viruses that can lead to cancer. Now, back when we covered the cell cycle, we know that all cancer is uncontrolled cell growth. And there's many different areas of regulation that regulates the division of our cells. And so essentially viruses damage cells. And so if these viruses damage the mechanisms that play a role in the cell cycle, viruses can lead to cancer. Okay, some common ones are HPV. In fact, usually as a young teenager, a lot of times maybe even starting around the age of 10. Nowadays there's an HPV vaccine. It's probably the most common STD. It's actually a wart virus, but it's been linked to cervical cancer. It's also been linked to oral cancers. Hepatitis B can lead to liver cancer. And really it's just the concept that viruses damage cells can become cancerous. So viruses could lead to certain cancers. Now, how do we treat viruses? Okay, now let's remember that viruses are not living cells. So if you have a viral infection, you can't just go to the doctor and get an antibiotic, because antibiotics are meant for bacteria. Now, you can get vaccinated, and most of you have probably had vaccinations. Maybe you get the flu vaccine. You could have gotten the COVID vaccine. You might have gotten the vaccine for HPV. Now, vaccinations are good for both bacteria and viral infections because what they do is they help prevent. Okay, vaccinations are pretty much preparing your body. So they're going to inject either live viruses or killed viruses or pieces of viruses into your body. Remember that your body has an amazing immune system and it's on high alert at all times. And so anytime it comes across something that it doesn't recognize, it's going to build up immunity to it. And so that's the purpose of the vaccine, is just to prepare your body. So just because you get a vaccine does not mean you're not going to ever have issues. Obviously, you could still get the flu, but hopefully it would not be as severe and last as long. Now, there are some antiviral drugs, but you have to understand that it's really hard. You can't kill something that's not living, so you can't just eliminate the virus. Normally the viruses, we just kind of have to treat the symptoms. Antiviral drugs might be used to kind of help control or make the symptoms not as bad. They may kind of block the actions of the proteins of the virus, but a lot of viruses, once you have them forever. Or you just have to rely on your body to fight them off, okay? And ultimately the cells, they get to the point where they can no longer infect other cells. But there's many viruses that once you have them, you do have them for life, like the wart virus, for instance. Herpes, a lot of your STDs, you just have to wait for them to rear their head and see some of those symptoms. So there's not always an easy fix. A lot of it is our own immune system. And that's why you can't just easily go and get medications to cure a virus infection. All right, the last thing we're going to talk about today are these other kind of biological weirdos. So remember that a virus is two components at least. It has genetic material surrounded by proteins. Now, these two weirdos are some other infectious agents, but they're not quite considered viruses. They're not living things, they're not bacteria, but they're not really viruses either because they don't have both components. Viroids are just little pieces of RNA. And for whatever reason, I don't know all the details or exactly how they work, but they're pieces of RNA that can infect plants. So they can be harmful to plants, they can affect their cells. But notice there's no protein. They're just pieces of RNA. Prions, on the other hand, notice it starts with PR. Prions are abnormal proteins, and they don't have the genetic component. But somehow these abnormal proteins can affect the brains of mammals. You may have heard of the term mad cow disease before. There was a big, huge breakout like in England. I want to say it was like the late 80s, early 90s. But what it does is these proteins become misfolded and abnormal, and then they trigger other proteins to become abnormal. There are some human equivalents, Kuru and the Creutzfeldt-Jakob disease. One of these was spread by cannibalism. So normally in order to kind of have this spread, there has to be the consumption of this meat. And so part of the issues with the cows, and they had to slaughter lots and lots and lots of cows because of this disease, but they were taking, like, after they processed the meat of the cows, they were taking the other parts and grinding them up and then using them for food. And so that's how these cows were kind of consuming this brain tissue that was damaged. For whatever reason, they affect the brain, which is going to cause odd behavior, which is how you get that mad cow disease. But there are human types. You don't really hear of it too often because you'd have to consume infected brain tissue, which is probably not very likely. But just know that these aren't classified as viruses because they don't have both the genetic and the protein component. They're one or the other. And that is where we're going to stop today. We're going to pick up with the finale of our notes here in a few days. Once again, welcome back, and I will hopefully be seeing you guys soon. Have a great day.